Hey everyone, my name is Alexandra and welcome to our series on pharmacology. In this chapter, we'll be discussing pharmacodynamics, which I like to think of as what the drug does to the body. We'll start with a discussion on protein structure and function, because many drugs work by interacting with proteins. We'll discuss a specific kind of protein called enzymes, and then we'll talk about how drugs interact with enzymes to cause a biological effect. We'll round off our discussion on enzymes with a practice question. Then we'll move on to discussing membrane proteins with a specific focus on receptors and how drugs interact with them, and we'll finish that off with a practice question as well. So let's jump right into talking about protein structure and function. Since we're focusing on pharmacology in this chapter, let's define what the term drug actually means. A drug is going to be any substance that has a physiological effect once it enters the body, and pharmacodynamics is the study of how drugs exert those physiological effects. Most drugs exert their effects by targeting proteins, which are macromolecules made up of amino acids. Here we have the basic structure of an amino acid, which contains a central carbon that forms 4 bonds. One bond is going to form to this hydrogen atom here. One bond is going to be made with this amine group, which is a nitrogen with 3 hydrogens attached to it. One bond is formed with this carboxylic acid group, which is the carbon with the 2 oxygens. And the final bond will form to this group represented by the letter R, and this R group is a variable group. And it's actually what defines a lot of the properties of the specific amino acid. For a more in-depth discussion of the different kinds of amino acids, you can check out the biochemistry video series. Now, amino acids are able to form bonds with one another. Specifically, the carbon atom from one carboxyl group is able to make a bond with the nitrogen atom from an amine group of a different amino acid. So over here we have 3 amino acids that are bound to one another. So if this here is the first amino acid, you can see here is its central carbon, here's its amine group, and then here is its carboxyl group. But it's missing one of its oxygens because this carbon is now bound to the nitrogen of the amine group of the second amino acid. And then over here at the end, we have the third amino acid, and it is also forming a bond with the carboxyl carbon in the second amino acid. This bond that forms between amino acids is known as a peptide bond. And a chain of multiple amino acids bound together with these peptide bonds is known as a polypeptide. Once a polypeptide contains enough amino acids that it begins forming more complex structures and serving a function in the body, it can be referred to as a protein. There's no exact number of amino acids that serves as a cutoff between a polypeptide and a protein, but most proteins are at least 50 amino acids long, although there are some that are shorter and some are much, much longer, all the way up to around 30,000 amino acids. We think of 4 different levels of organization when considering the structure that defines a protein. Primary structure is the linear order of amino acids that are connected by those peptide bonds. So for example, you could have a protein where the first amino acid is a lysine, the second amino acid is a valine, and the third amino acid is a histidine. And recall that the identity of the amino acid is defined by its unique R group. The primary structure of a protein is going to be held together by rigid peptide bonds, which are these bonds that form between the amino acids that we discussed earlier. Now, if you have a long chain of amino acids all connected to each other, it's not going to be very energetically favorable for this molecule to just exist in a straight line with no intermolecular interactions. Instead, what will happen is the atoms that form the peptide bonds will form hydrogen bonds with one another. And that ends up giving rise to periodic protein folding motifs that form the secondary structure of a protein. The 2 most common types of secondary structure are alpha helices and beta pleated sheets. Alpha helices form when the oxygen on the carbonyl atom forms a hydrogen bond with a hydrogen on the nitrogen atom on an amino acid 4 groups further down the chain. So here on this strip of paper, I've drawn out a representation of the primary structure of an amino acid that contains 5 amino acid groups connected by peptide bonds. An alpha helix forms when this amino acid forms a hydrogen bond with this amino acid, and it causes a twist in the protein like this. And then if we imagine that this protein contained more than 5 amino acids, it would eventually form this helical shape, and that's going to be your alpha helix. But remember also that amino acids themselves are three-dimensional structures, so the R groups of the amino acids when the alpha helix forms are actually going to be pointing out from the helix, so they'll be pointing out this way. The second most common type of secondary structure is the beta-pleated sheet, which occurs when different strands of amino acids line up next to each other and form hydrogen bonds. With our strip of paper example, the secondary structure would look something like this, where this amino acid is forming a hydrogen bond with this amino acid, causing this interaction. And in this secondary structure, the R groups are pointing out from the plane of the secondary structure. So they're pointing out towards you or towards me. When I was learning about this for the first time, I wondered if both alpha helices and beta-pleated sheets are both formed due to hydrogen bonding between atoms involved in the peptide bond, and peptide bonds are the same between all amino acids, why are there even different kinds of secondary structure to begin with? And the reason is differences in primary structure. Specifically, the order of amino acids defined by their unique R groups predicts what kind of structure can form. In the folding of the alpha helix, the R groups wind up really close to one another. And that means that amino acids that have really large side groups, such as tryptophan and tyrosine, or amino acids with like charges, like lysine and arginine, are going to have a hard time folding into alpha helices because those R groups aren't going to want to be close together. Additionally, the amino acid glycine, where the R group is just an additional hydrogen, is much smaller and more flexible than most other amino acids, and that often leads to a disruption of secondary structures like the alpha helix. Another amino acid that disrupts alpha helices is proline, and the structure of proline looks like this. Proline is unique in that its R group bends around to bond with the amine group of the amino acid, which means that this amine group can't participate in this hydrogen bond. Also, because of this unique binding, proline ends up having a unique shape compared to the other amino acids, and it actually creates a bend in the secondary structure of the protein, which isn't compatible with the folding of the alpha helix. So if you have a primary protein structure that contains either large R groups, like charges next to each other, glycine or proline, an alpha helix is going to be destabilized, and you're either going to get a beta-pleated sheet or a stretch of unstructured protein. Once the secondary structures are formed, the protein is going to continue to fold even further into the tertiary structure. And this is represented by this diagram here. So within this tertiary folding structure, there is going to be the secondary structures we just talked about. Maybe there's a region of an alpha helix over here. Maybe there's a region of beta-pleated sheet over here. And this is all going to fold together into a three-dimensional structure. The folding of the structure is mostly driven by interactions between the R groups of the amino acids, unlike secondary structure, which was driven by hydrogen bonding between the peptide bonds. Predominantly, this folding is driven by hydrophobic interactions. So proteins are going to contain both hydrophilic and hydrophobic amino acids. Let's say that this protein is folding in the cytoplasm, as many proteins do. The hydrophobic amino acids are going to want to fold into the center so that they are hidden from that aqueous environment. The hydrophilic amino acids are going to want to fold where they can interact with the aqueous environment of the cytosol. But depending on the identity of the R groups, you can also have hydrogen and ionic bonding forming, as well as disulfide bridges, which can form between two cysteine amino acids. For many proteins, folding into their tertiary structure is enough to render them functional. However, some folded polypeptides are subunits that need to assemble with other folded polypeptides in order to form that fully functional unit, and this is known as the quaternary structure of a protein. For example, this quaternary structure here represents hemoglobin, which carries oxygen in red blood cells. And hemoglobin consists of 2 alpha subunits and 2 beta subunits. Each of these subunits folds independently and forms their own tertiary structure before coming together to form the quaternary structure that is functional hemoglobin. The subunit interactions that make up quaternary structure are driven by the same types of intermolecular forces as tertiary structure. Understanding how protein structure forms is important because the structure of a protein defines its function. For example, fibrous proteins involved in structural support like keratin and collagen have primary structures that allow them to form long, uninterrupted alpha helices. On the other hand, proteins that need to be able to interact with a smaller molecule like hemoglobin or many enzymes need to be more globular, which means they need to have a higher amount of unstructured, flexible regions. Proteins have a ton of diversity in their structure because there's 20 different kinds of amino acids, and they can be combined into a primary structure in any order. Because there's so much diversity in the structure, proteins are able to serve lots of different roles in the body. Some of the major functions of protein involve providing structural support, like collagen, participating in cell signaling, like this receptor protein here, putting molecules between cells, like hemoglobin, and catalyzing important biochemical reactions. Let's summarize what we talked about in this video. Proteins are a macromolecule made up of amino acids, and they serve a wide variety of important functions in the body, which is one of the reasons they make such great drug targets. The function of a protein can often be predicted by its structure. There are 3 to 4 levels of protein structure. Primary structure is the linear sequence of amino acids that are connected by peptide bonds. The primary structure defines what kind of secondary folding structure, which is commonly either an alpha helix or a beta pleated sheet, is able to form. This secondary structure forms through hydrogen bonding between the peptide bonds of the amino acids. The overall 3D structure of the protein, which is known as the tertiary structure, is driven mostly by interactions between the R groups of the amino acids. And then some proteins will also form a quaternary structure, where fully folded subunits will assemble to form a fully functional protein. When properly folded, proteins serve a variety of critical functions in the body.